Hello everybody. Sexual reproduction and genetic variation covers the second part of Chapter 2 of Pearson's genetic. Before we will go over meiosis, I will ask one major question. Why do we need sexual reproduction? Why cannot we all reproduce by mitosis? And the answer would be very simple. Do you recall mitosis creates clone, identical individuals? So if we all reproduce by mitosis, we will be all identical, all alike. Not only it will be very boring, but it will be dangerous. Imagine that we will have the same sensitivity to the same pathogen, susceptibility to the same diseases. So what disease outcome, and we will be wiped out. So sexual reproduction, from answering this question, you already can deduce what is outcome of sexual reproduction. It is a genetic variation. The sexual reproduction introduces genetic variation. Genetic variation is base for natural selection, which is a driving force behind evolution. So as you can see, the evolution come up with very important mechanism by which it can create the base from which it can select the best suited genotypes and phenotypes. Now, how does it happen? So, sexual reproduction requires fertilization, which means fertilization is fusion of haploid gametes. Gametes refer to eggs and sperm. So, in order that fertilization can take place, the gametes has to be created or processed having only haploid number of chromosomes, meaning 1/2. And therefore, sexual reproduction actually proceed in two parts. First part of sexual reproduction is meiosis. Meiosis is a mechanism which produces haploid gamete that have only one set of chromosomes, so they can fuse together in second step fertilization, and basically this fusion reinstall diploid full number of chromosomes. So let's talk now about meiosis. Okay, so let's talk about the mechanism of meiosis. But first, let's point out to the major difference between mitosis and meiosis. So what is the outcome of mitosis? So when we talk about mitosis, you recall one cells is divided and produces two daughter cells with diploid number of chromosomes which are identical, which are identical to each other and identical to the parental cells. That's mitosis. In meiosis, basically outcome of meiosis is that it creates 4 different cells in 2 different divisions, meiosis 1 and meiosis 2, and these 4 different cells are variable, they differ from each other, and they do have only haploid number of chromosomes. So in order to proceed from diploid to haploid number, meiosis 1 is actually first part or so-called reduction division, where diploid number of chromosomes is reduced to haploid. So outcome of meiosis 1 are 2 daughter cells. Each cell has haploid number of chromosomes. However, each chromosome still composes of 2 sister chromatids. So in order to separate sister chromatid, we need meiosis 2. And in meiosis 2, the sister chromatid separate You see one chromatid, second chromatid in daughter cells. So each of these daughter cells give rise to 2 additional cells. So outcome are 4 cells, each one with haploid chromosomes, haploid number of chromosomes, and each chromosome consisting just one single chromatid. Okay, so let's go over the mechanism of meiosis, not the detailed ones which are outlined in second animation in Launchpad. However, let's pick up the most important differences outlined in meiosis 1 and meiosis 2. Firstly, remember that like mitosis, meiosis is preceded by an interphase step that includes G1S and G2 phase. So before cell commits to produce gamete through meiosis, it already replicated chromosomes, so each chromosome consists of 2 chromatids. Now, what are the major differences? Prophase, metaphase, and which outlines anaphase 1. So during prophase of meiosis 1 is relatively long, and it comprises of several stages: leptotin, zygotin, pachytin, diplotin, and diakinases. What happened during these stages? The homologous chromosomes align and form pairs. They form so-called tetrads. Tetrads because they are composed of 4 chromatids. These tetrads are held together by synaptonymal complex, which is appearing during pachytin stage. During prophase, during pachytin, and diplotin, basically chiasmata start to appear. You can see them very well in diplotin stage. What are chiasmata? Chiasmata are crossing you can observe in chromosomes where crossover between 2 chromatid, non-sister chromatid took place. We will talk about that. So what is that crossing over? We will come to it. It will be actually during prophase 1 if you have 2 homologous chromosomes with 2 different alleles. Let's say here is one which has dominant, one recessive. Basically, the crossing over triggers breakage and repair and exchange of pieces of chromosomes. So after crossing over, you will have 1 chromosome which would have recombined allele, and second chromatid which also would have recombined alleles. So keep in mind, crossing over is the first mechanism by which meiosis introduces genetic variation, and we will come over, and it takes place in prophase 1. Another difference, if you compare mitosis and meiosis, is metaphase plate. You recall in mitosis, if you take mitosis and you look in metaphase plates, chromosomes are aligned side by side. Okay, while in meiosis here, homologous chromosome align side by side. This random alignment, we will talk about it, is second mechanism which introduces genetic variability, and it's typical for metaphase 1 because 2 homologous chromosomes are aligned at metaphase plate. And this is important because kinetochore, centromere of each chromosome, is connected to spindle from coming from the opposite pole. So basically during anaphase, when the spindles are shortened, basically it's whole chromosome which are pulled in different poles. It's not chromatid which are separated during anaphase 1. Those are chromosomes which are separated. Now, if you go to mitosis and meiosis comparison, so you see major differences prophase. In meiosis, there is crossing over. Metaphase plate where two homologous chromosomes align side by side during meiosis 1. And this is base for separation, segregation of the chromosomes, whole chromosomes during anaphase 1. So outcome of meiosis 1 are two cells, each one with haploid number of chromosomes. However, each one has still two sister chromatids. So meiosis 2, which follows meiosis 1, assures that the sister chromatids are separated to daughter cells. Look at the metaphase plate during metaphase 2. What does it resemble? It resembles mitosis. So what does it tell you? Chromosome, that haploid number of chromosomes now align in metaphase plate, and sister chromatids separate to daughter cells. So if you compare mitosis and meiosis 2 and meiosis 1, you will see that mitosis and meiosis 2 have certain similarities. If you look at the outcome of meiosis, you will see here 4 cells, each one with haploid number of chromosomes, randomly distributed. Homologous chromosomes are randomly distributed, and the genetic variability might be also introduced by crossing. So let's talk about crossing over, the first mechanism of generating genetic variation. Crossing over takes place during prophase 1, and what does happen is that allele located on non-homologous chromatids of homologous chromosome pair are exchanged. Let's look at this scenario. We have one pair of chromosome here. Let's consider red one coming from mother and blue one from father. We see two genes, gene A and gene B. Now, mother possesses both allele from both genes as a dominant, while father carries both recessive alleles. Now, what happened with these alleles during DNA synthesis? You know, during DNA synthesis, DNA is replicated, and we end up with two identical copies of DNA for each chromosome. So, basically, if imagine that crossing over doesn't happen, what would be outcome without crossing over of meiosis following meiosis 1 and meiosis 2? You will end up with 4 daughter cells. Each one would carry combination which you find on parental chromatids. So we will have 2 gametes with both dominant allele A and dominant allele B, and 2 gametes which would have both recessive allele A and allele B. So this would be no crossing over. This will be gamete which would be created without crossing over. So what happened during crossing over? You remember during prophase 1, homologous chromosomes form tetrads. They come in close contact, and there is a breakage and rejoining of pieces of chromosome and exchange of chromosomal pieces, which actually would shuffle alleles between non-sister chromatids. So what would be outcome? You will still have, if we have crossing over, single crossing over between 2 non-sister chromatids, you still end up with half of the gamete which would be non-recombinant. You see, they would have parental allele. One would have both dominant allele A and allele B, and another would have both recessive allele A and allele B. However, half of the gamete would be recombinant as a result of crossing over of the allele shuffling, and one of the recombinant allele would have recessive allele A and combined with dominant B, and second one would have dominant allele A combined with recessive allele B. So this mechanism of genetic variation relies on shuffling or exchange between two non-sister chromatid on one homologous pair. So what does it mean? It changes combinations within one homologous pair of chromosomes. Random chromosome distribution is the second mechanism of generating genetic variability during sexual reproduction, and particularly referring to meiosis. So let's look at the cells with three homologous pair of chromosomes. Let's follow the same scenario as before, and let's look at red chromosome as those coming from mother and blue from father. Okay, so you recall DNA replication assures that each chromosome now is made of two sister chromatids, meaning two identical molecules of DNA. Well, if only exception here can come if there was a crossing over during prophase 1, so they would not be identical. Keep it in mind. However, let's consider first here that there was no crossing over, so we have identical chromatids. Now, what happened with this chromosome, homologous chromosome, during metaphase 1? You recall during metaphase 1, homologous chromosome align side by side in metaphase plate. Now, how do they align? It's completely random. There is no rule which would predict or dictate whether mother chromosomes will go on one side and father or another, or whether they would do different combination. So any combination which is outlined here is completely random and has the same chance to happen. So the probability of each combination is equal. So what does it mean? That basically we can come up with different gametes with different combination of chromosomes, mixed and matched from mother and father, meaning if you receive this gamete from your mother and different gamete from your father, let's say, so for you this chromosome would relate to your grandmother. So in that case, if you get this gamete, there is very high chance that you will resemble your grandmother. If you get this gamete, respectively, if you are formed basically on by gamete, by fusion of this gamete from your mother and gamete from your father, you will resemble your grandfather. So now, how many combinations we can have? It always depends on number of chromosomes. So firstly, how do we calculate the number of combinations? You see, if you have organism which has only 3 pairs of chromosomes, 6 chromosomes, so you can have 8 different gametes. So how do we calculate it? First, the root of this calculation formula is 2, and it is because you always have 2 chromosomes aligned side by side, and you expand it to n, where n equals number of haploid chromosomes or number of chromosome pairs. So in this case, which is outlined here, it would be 2 expand 2, number of pair is 3, 2 to 3, which is 8. Now, can you imagine in humans, if you just forget crossing over and you focus only on random chromosome distribution, and we have in humans 46 chromosome, which comes in 23 pairs. So the formula would be 2 to 23, which gives you combination X combination, which we can form during gamete formation. So this second mechanism has power to just shuffle chromosomes and mix and match chromosomes from mother and father and come up with different combination. Now, if you follow the combination of the, imagine that each chromosome is a vehicle for different genes. So the shuffling brings a new gene combination to gamete. So during fertilization, basically two gametes, one from father and one from mother, are selected randomly and fused. So this is a third mechanism of generating genetic variability when two unique gametes actually fuse and form zygote, which would divide and form embryo. Now, you recall we talked about first crossing over and second random alignment, which both contribute to this unique gametes which randomly fuse during fertilization. So what is the difference? Crossing over shuffles allele which are localized on the same chromosome pairs. So let's say you look at the gene localized on locus on chromosome gene A, which is localized on chromosome number 1. So basically, the shuffling via crossing over refers to the same gene which just shuffle allele between non-sister chromatid on the homologous chromosomes. Random alignment shuffles alleles which are on different chromosomes. So it makes a new combination of different allele of different genes. So what are consequences of meiosis? First, one parental cell, which is diploid, give rise to 4 daughter cells, which are all haploid. You remember meiosis 1 and meiosis 2, so the number of chromosomes is reduced to half during meiosis 1. Now, not only these 4 cells are haploid, but they are also genetically different. And then this genetic difference is introduced by crossing over and by random chromosome separation during as a result of random alignment during metaphase 1. So this variability in gametes, imagine if you have 46 chromosomes, 23 pairs in humans, how many different combination of allele of different genes and exchange due to crossing over are possible. Now imagine that this combination take place in the egg formation and also in sperm formation, and there is a random fertilization. So all this assures that outcome of meiosis is formation of haploid cells with high genetic variability. So let's go back again and compare mitosis and meiosis. There are differences in the mechanisms how mitosis and meiosis proceed, but what I would like to strengthen or stress the most is outcome. Mitosis creates clone identical cells. Meiosis creates cells which are variable, which are different from each other. The alleles, combination of alleles is different in each daughter cell. So meiosis is the base for introducing genetic variability in population, which is base for natural selection and for evolution. So now let's compare the most different stages. You recall prophase. Prophase 1, you have formation of tetrads, crossing over taking place during prophase 1 of meiosis. First mechanism of introducing genetic variability. Second difference, look at metaphase plate. During metaphase of meiosis, chromosome align side by side. In metaphase 1 of meiosis, homologous chromosome align basically next to each other in metaphase plate. What is the outcome? What is the outcome of this? In mitosis, during anaphase, sister chromatids separate, and each sister chromatids go to different cells. This would be one cell, this would be another. What happens during meiosis 1, it's chromosomes which separate. You see, each chromosome still have 2 chromatids. So basically, the separation of chromosomes instead of chromatids assures division of chromosome to half, or reduction of number of chromosome to half. Now, the major differences, mechanistical differences you find during mitosis and meiosis 1. If you compare mitosis and meiosis 2, you will see that there are many similarities. But I stress again, the major difference is outcome. Mitosis creates clones, while meiosis creates genetically different cells, different gametes. So, looking on the differences between mitosis and meiosis, one question comes up to my mind or to your mind. Why sister chromatids separate during mitosis anaphase, however they do not separate during anaphase 1 of meiosis? So we have to explain this, we have to go back a little bit to interphase, to S phase. You recall during S phase, sister chromatids are synthesized, respectively. DNA replicates and forms sister chromatids. Now, at the same time, protein complex called cohesins are assembled and hold together sister chromatids. When during metaphase, chromosome align in metaphase plate, and basically spindle assembling checkpoints assures that each chromatid is attached to proper pole, to proper spindles. This successful pulsing spindle assembling checkpoints activates enzyme called separase, and separase would break down cohesin and release sister chromatids, which can now travel to different pole of the cells. So what about meiosis 1? Why it does not happen during meiosis 1, or does it? Actually, it does. Separase is activated also during meiosis 1 anaphase. However, it only breaks cohesin at the arms of sister chromatids. At centromere, cohesin is protected by particular complex of protein called shugoshin, which in Japanese means guardian angel. So shugoshin protects cohesin from action of separase and enables chromosomes stay together, meaning sister chromatids stay together and travel together to one pole. Now, after anaphase 1, shugoshin is broken down, and therefore, during anaphase 2, cohesin is not anymore protected and can be broken down and allow sister chromatid, each one, to travel to different places. So remember, function of cohesin and shugoshin in the chromatid alignment and separation. So now let's talk where we encounter meiosis respectively when during our life cycle cells reproduce by meiosis and where by mitosis. Keep in mind that meiosis is employed only when gametes are produced during spermatogenesis and oogenesis. Once sperms and egg are fertilized, they fuse, and fertilization produce zygote, which is diploid. Following formation of diploid zygote, all embryonal cells reproduction, division is done by mitosis. Equally, once we are born, all cell reproduction takes place through mitosis. So meiosis is only employed during gamete formation. Now you have here outline of spermatogenesis and oogenesis. You can see a lot of similarities. Formation of spermatogonium, oogonium, primary spermatocyte, primary oocyte, secondary spermatocyte, secondary oocyte, ovum, and sperm. So you will see there are many similarities in both processes, but there are also differences. First one is the outcome. Male gametogenesis, spermatogenesis form for sperm, while female produces only one ovum. During each division, meiosis 1 and meiosis 2, only one of the daughter cells acquire whole cytoplasm during cell division, during cytokinesis. The second one forms a polar body which later on disintegrates. The same happens also during meiosis 2, so the outcome is only one egg. Reason for it is that eggs requires a lot of nutrient to support embryonic development before placenta forms. So this is just very basic difference. There is the major difference in timing. Spermatogenesis is initiated during puberty in males and continues throughout the adult age, continuously goes from meiosis 1 through meiosis 2 and sperm formation. So it's a continuous process. During oogenesis, there are not many, but several stop and restarts. Oogenesis is initiated during embryonic development and is arrested at prophase 1. So, female is born with certain number of primary oocytes which are arrested at prophase 1. During puberty, every month when there is a hormonal change during and after puberty, this hormonal change triggers completion of mitosis 1 and formation of the ovum, which is ovulated every month. However, this ovum completed only meiosis 1. So when is meiosis 2 completed? Only upon fertilization. So fertilization triggers restart and completion of meiosis 2. So sometimes it can take 20, 30, 40 years from initiation of meiosis 1 and completion of meiosis 2. And this time lapse and stop and restarts have implications. And those implications, I strongly encourage you to go and read a science article from Science, which is called Sex Matter in Meiosis. It's very interesting, and it will explain you what can be actually outcome or what might happen during all these stop and restarts. Okay, so we covered chapter 2, so in next recordings we will go to basic principle of inheritance. Before we do so, I would like to stress again, please make sure that you understand meiosis and it links to genetics, genetic variation, and behavior of chromosomes. I would repeat again that chromosomes are vehicles which carry genes from parental cells to daughter cells. So if you understand behavior of chromosomes, you would understand inheritance, and you will understand Mendelian's law of segregation and independent assortments.